Okay, so first of all, thank you very much for the opportunity to present MGnify and how we're expanding into the viral annotations. As Joanne mentioned, I'm from the European Bioinformatics Institute, and also a little disclaimer up front, there are going to be many things that I say that are very similar to the things that Simon has just said, but we have not colluded on our slides. So just to give you a bit of an overview, so MGnify is a free-to-use resource for the archiving, and that's very important. And in fact, we have a link to INSDC, so the European Nucleotide Archive, for the archiving, assembly, analysis, and browsing of microbiome-derived sequence data. That's a bit of a catch-all. What that means is if you've got a shotgun metagenomic data set, you can upload it to us. We will help broker that into ENA. We can then assemble it, broker those contigs in, and then we will then pull those back out and analyze those in a systematic way. We are very keen to make sure that our pipelines are reproducible, and I'll touch on this later. And then we have lots and lots of different visualizations. But I really wanted to start off with a graph here showing you the number of, showing you the growth of assemblies that are in INSDC. So in this particular slide, along the x-axis, you have the time, the year, and along the y-axis is the number of submissions that have entered INSDC. So the shaded gray area shows the totality of all assemblies. If you look and pick up the blue line, that represents the numbers of submissions that correspond to isolate assemblies. Whereas in the green line, the solid green line shows the totality that's come from environmental samples or metagenomics. The broad dashed line shows the number have come from MGnify, and the fine dash from the community. And what you can see is both the community and MGnify are submitting it into this. One of the things that I quite often get asked about this slide is why is the flat line in MGnify? And the reason is that actually initially INSDC were accepting our assemblies in a particular route, and essentially you seeing that we were doubling the size of the numbers of assemblies. And so here what we had was the fact that actually we started causing all sorts of problems across both ENA and INSDC. And so we have had to back out our assemblies and reposition those. And this is a bit more important, and I'll come to this at the end of my talk about why this is important and important to start thinking about this. So why am I telling you all about this? So, and I'm going to talk to you a little bit about prokaryotes. So one of the things that we do in my team, so I have the service side that provides MGnify, and I also have a research team. So what we do is we have a synergy between those two parts of my overall team. And one of the areas that's come from my research group is the fact that we've been studying the human gut microbiome. So this is, you know, again, this shows you the sort of orthology between what MGnify has and JGI, and the fact that we are sort of have that gut microbiome data. So we started off assembling 13,000 human gut microbiomes. We established there was about nearly 2,000 novel species in there. So these are the metagenomes, assembled genomes. These are prokaryotes I'm talking about at the moment. And there were two other papers at the same time, one from JGI and another one from Nicholas Agata's group, and we amalgamated those to form the Unified Human Gut Genome Collection. Really, the point of showing you this side is that in that bacterial space, we tapped into huge amounts of novelty. So 80% of those mags are yet to be cultured and could be considered as novel. And that corresponded to over 210,000 genomes that we were handling. And so that's about the size that the number of genomes, isolate genomes that are in NCBI. So this is fairly substantial, but it also illustrates the novelty and of generating prokaryotes. We're generating more and more MAG collections, these metagenomes assembled collections, where we actually go into individual environments and pull them out. So we have a skin collection that's come from the research team, and we also have another data, another collection coming from chicken cecum microbiome. We have these coming from all over the place. One of the characteristics of all of these is the novelty that we have in there. So on the previous slide, I should have pointed out, in fact, the green is the novel color. So coming off the tips of these phylogenetic trees are the novel mags. On the skin microbiome, we actually find there's not so much novelty, but you still do find a certain amount. And when we go into the chicken cecum, and this is the color coding shown on the outside in this particular case, the green, we still see a lot of novel genomes that are coming out of the cecum. So there's lots and lots of novel bacteria that we can pick up on. We were also wanting to just illustrate the fact that we have this data life cycle, and the fact that we constantly are trying to make sure that when we produce an assembly, it goes into ENA. When we generate the bins, they go into ENA, and then the mags go into ENA, and then MGnify produces reference catalogs for these prokaryotic genomes, where we remove all of the redundancy, because the scale is pretty big. And it's very important. We've been very mindful of this, because what we want to do is have the complete provenance of the data of how all of these genomes came about. So why am I telling you all of this at a Viromics conference? And the reason is that we care about all of these. We started with the prokaryotic genomes, but we have all of these assemblies. So we have gone back into the gut microbiomes to say, okay, what else do these metagenomes and genomes from the human gut microbiome contain? So this work was done in collaboration with Trevor Lawley's group. He focuses a lot in isolating and culturing bacteria from the human gut. And so we were able to establish a pipeline where we were taking both isolate genomes and metagenomes. And then we used a tool called Verify, which combined at that time the outputs of VirFind and VirSorter. And we screened millions of contigs to then try and identify those that were of viral origin. There was a length filtering, so we only allow things greater than 10 kb to go through, and a neural network. And that neural network was designed, it was trained very much on existing data sets that are definitely known to be viral, and also things like ICEs, so these integrative conjugative elements, to try and make sure they look like viruses, but they're not viruses, and trying to build up features that could allow us to distinguish. Going through that process, that allowed us to establish the gut phage database, where there we had over 142,000 high quality genomes of human gut bacteriophage. Now, the vast majority of these pass the check V criteria and have next to no detectable contamination. And I use a caveat with that. So what we actually find is that this is a very, very high quality resource. It's currently only available on the FTP site, but this is where MGnify is moving to. I want to just present you just some of the anecdotal findings from this particular study. So when we take those 142,000 genomes, we then compare those viral genomes back, and we can test the prevalence of those. What we can actually ask is, okay, out of the clustering of those contigs, which is the largest? Now, the crass phage is well known to be highly prevalent, and that was our first and our most dominant cluster, as indicated on the left-hand side of this slide. But the next one down, which is still found hundreds of times, was something called the gubophage. Now, this had not been noticed before. And when we start then looking at the prevalence of this in different continents, what we find is that actually Europe, it's most common in Europe, Asia, Oceania, then North America, Africa, and Southern America descending down. But one of the points is you have to sort of balance that out with the relative numbers of samples coming from there. So actually North America seems to be depleted in the gubophage, and the rest of the world, it corresponds to roughly the numbers of metagenomes that actually went into that analysis. But the take-home message is this phage is highly prevalent. It's in the human gut, which has been relatively well studied, but nobody's noticed it before. So we've looked at we did a lot of work to make sure that actually this goober phage was an extension of the crass phage. And this really sort of illustrates on the left-hand side that this was quite distinctive. So we build a phylogenetic tree of those. This definitely represents a distinct clade. And then within that, we saw two major groups within the goober phage. And one of the groups we could subdivide into other groups. Now, this is one of the challenges in terms of when is something a viral species, a subgroup, etc. I think this is one of the challenges we face, but at least we could do this grouping and we could start having an idea of how this goober phage could actually be broken down. So we took the whole of the gut phage catalogue, and then we asked, which environments do we actually find this in? And so this is work in collaboration with the group that has established the Seratus tool. And what we did is we took the gut phage database, the terminase as a marker, and then scanned a whole bunch of metagenomes for the presence of this terminase gene. This phylogenetic tree is shown shows that tree based on that terminase gene. Around the outside are different environments. So you've got green, human gut, manure in black, red is mining, blue is aquatic, and then on the outside is RESI, just showing you where you can actually pick this up. And what we find is actually there are some big gaps. So there's lots of cases where human gut and manure, that's not really surprising that they've got a very similar pattern, but there are some gaps. So that's sort of 5 o'clock on this phylogenetic tree. There are cases where there seems to be a phage that seems to be very specific to the human gut, but there are also areas where we actually find that across multiple biomes, and you find it in aquatic environments. Now, you could say that aquatic environments could have human fecal samples in them, but possibly not quite at the rate that we were establishing it. But that's still something where it's not conclusive that these are found in the environment, but we do have evidence to start suggesting that may well be the case. You can find many of the human gut phage outside of the human gut. So I wanted to talk a little bit more about this Verify pipeline, and this is a very brief overview. I'm not going to go through this whole process, but there's essentially a standard sort of set of steps that we go through for this Verify pipeline. This is a standalone pipeline that you can run yourself as well as running it through MGnify. And we start off with an initial bit of standard QC and of the contigs that have come from a MGnify assembly. We then run through VirFinder, VirSorter, and PPR Meta now, and then we combine the set of those and try and establish high confidence, low confidence set of contigs, and then we go through and annotate those. Now we've got the whole repertoire of what you can do in Verify. You can switch on comparisons against IMG-Vir. You can compare it to other databases like the Viromics, so the VFogs, but we have our own database called the Viromics, and I'll talk about that now. So this is a specific database that has been established by another group. So this is by Alejandro Reyes from Colombia. His group has just published this catalogue of Viromics. These are 31,000 profile HMMs that are able to identify based on viral genes that can provide taxonomic annotations. And what we've done is take those. So on the right-hand side of this slide is showing you the distribution of the known virus world and how well that those Viromics model that space. So we can detect both prokaryotic and eukaryotic viruses, and we have reasonable coverage. And coming off the edge is the number of times a number of those Viromics that actually match that. So some areas aren't great, and that's purely because of the fact that there's not many sequences to build a sensitive profile HMM in that space. And so not having enough examples of viruses is a key limitation. The graph at the bottom here shows you the number and the limitation in terms of what level or the finest grain level that these Viromics can annotate. So some are very, very coarse grained and only annotate family or order level, but some give us sort of genus specific annotations. So what we do is we run those VIRFOGs, and then we annotate those high confidence and low confidence, and then we have some cases where we might have clashes of annotations on a config, and the whole right hand side is to try and actually have a system of actually having a voting system that allows us to try and identify. One of the things that we've introduced into the VIRFOGs is actually comparing those against NCBI database and actually trying to identify individual per model thresholds that allow us to indicate whether something's a significant match or not, to really try and get to that taxonomic level. What we found is we couldn't use a standard E value for the profile HMM because every model has a slightly different sensitivity. And that's the same sort of philosophy that many of the protein family databases have taken, like PFAM. So finally, with Verify, you have an output that allows you to do some visualization. As I mentioned, this is sort of a standalone pipeline that you can run yourself, and it's been integrated into the MGnify assembly analysis pipeline. And that will shortly be rolled out into production, where we'll reanalyze all of our 30,000 metagenomic assemblies. We're also adding the CRISPR-Cas finder, such we can try and start linking hosts back to phage, and that will allow us, and also combining with this with the MAG catalogs, will start allowing us to start building this network of relationships between environment, hosts, and viruses. Our workflows, just as a slight aside, we try and make sure that they are all formally described. There are many different workflow languages. I do not intend to go through what this means in terms of a workflow, but what it does is it gives us full provenance of what we've done. So the tools we've used, the parameters and the reference databases, and you can actually get these workflows from Workflow Hub, which is a fairly new way of actually publishing your workflows, and we have both a Nextflow version and a CWL version of Verify. So as a final example, we keep having all of these different catalogs. We have a salmon gut microbiome, and this was really just to illustrate a couple of things. So this is a little bit of work. It's quite preliminary, but it still illustrates a quite nice point. So we've analyzed a salmon gut metagenomics data set, and what we find is, although we can identify tens of thousands of potential viral contigs, 98% of those are unclassified with Verify. And if we look for other classification systems, the number of unclassified goes up. So, you know, there's 2% that we can actually do something with. One of the other things is we have this notion of high quality and low quality in Verify. We've done some sort of comparisons to check V. And really the high quality ones are the ones that, you know, we would be starting to think about having into a catalog in MGnify. So this is something that we're working on. So we've got lots of these data sets that are available, and we're not quite there to having this full database, but we're very, very close. And so this really just illustrates the fact that, you know, we're not trying to check V and Verify aren't trying to compare like for like. So you can't just say, okay, they're completely wrong, they're misannotating. But the concept of what things are good, what we can see is that out of the 40,000, we're only having a few hundred that are actually, we think, are good quality viruses. And I think that's one of the key challenges facing the community. And also what you'll see from this corona chart is that the taxonomic resolution is still not wonderful of these diagnostic HMMs, especially when you go into environments which aren't well studied. So I started off talking about the prokaryotic world, and you might be slightly turned off by that, but I'm sorry. But the reason for doing this is that this pushing back into ENA is a very key part of our remit of making sure that everything's archived. Now, for the prokaryotic world, as I mentioned, we started causing issues with INSDC. So new layers were introduced where we would start pushing metagenomic assemblies, metagenomic bins, and mags. And this was really to reflect that these different levels were, as shown on this slide, we'd start off with raw reads, we would submit an assembly which would reference that raw reads, and then we would form bins, this collection of all potential bacterial genomes, which would reference that assembly, and then the very good quality representatives would go into this mag layer. Now, we've been working with our ENA colleagues as part of this to be thinking about, okay, how do we do this for viruses? And this is something that I think we would welcome feedback on. Now, we're having an internal discussion about what level of quality would require a corresponding level. So just to illustrate, what we're currently thinking is making such that you have the same assembly, and then you can point to essentially viral contigs that are partial, and you submit them with the output from your MyUvig results. But then the big discussion is this UVig, the MAG equivalent level. So this would be all contigs that come from a metagenomic analysis. Then this UVig is something where you know, this is where you want to assert high quality. Now, ENA are trying to push for finished, and I have the full definition if we want to go into what that is, but this is basically meaning circular, or having, as Simon said, the repeats at the end so that you can make it circular. I would be arguing this 90%, the check V level, but it would be interesting to hear the community's feedback on this. But what we want to try and do is, as people are doing all of these analyses, is capture this in the layer such that, you know, we can go back and then go looking for things, but not necessarily having to analyze the millions of viruses that are going to be out there. So just to really sum up, in MGnify and the team, we've got multiple virus catalogs established. There are papers either already out, in press, or in preparation. The moment that data is all limited to our FTP site, I apologize. But where we are is the fact we have this pipeline that is already available for use, and it's established, and it's already wired into our production systems. We're just waiting on the release of some of these prokaryotic genome catalogs before we start on the next step. I blame the pandemic for that delay. It's caused some issues, some disruptions to life. But one of the things that we do want to do is actually, when we roll this out, is having this linkage of environments and host wherever possible. And this information will initially be on the analysis level, so on a metagenomic assembly. As just alluded to, ENA is establishing these archival layers. We welcome feedback on that. With then the next step, MGnify having the viral catalogs that will be in parallel to these prokaryotic MAG catalogs. As with all these things, there are many people who have contributed to the work. The people in bold in my team, particularly down the left-hand side, Alex Almeida, Martin Baroccacher, Varsha Kale, and Sarah Kashaf, and then everyone else who has contributed to this work, especially Alejandro Reyes for those VirFogs, and Guillermo Rangel, who really did a lot of the prototyping for the Verify system with Martin Holzer. And thank you for listening, and I can take one or two questions maybe.
Excellent. Thank you, Rob. That was a wonderful talk. So yeah, we have time for one question, and we'll ask this one from the group. Can one include another viral identification tool such as Vibrant within the Predict virus as part of Verify, and how easy or challenging would that be?
So in theory, yes. And how challenging would that be? To introduce it into the workflow, it would be relatively simple. So we already have other tools or other reference databases. And so it would just be a question of that workflow is quite logically laid out, and it is a human read. I can read it. Okay, I may not be normal, but I can read it. And so it is readable. And so you can actually template off these tool descriptions, then drop it into the workflow. And it actually runs very easily to extend this. And this is how we do a lot of evaluation of new tools that come out. So for, OTU definitions for amplicon data, we can test tools and we can swap them out very easily. So it wouldn't be, terribly difficult. One of the things that we are doing it to verify is adding in check V, and the amount of work that is actually relatively simple.